Hello, everyone. Welcome to today's lecture on genetic disorders, which is a foundational topic for both your university-level genetics understanding and also highly relevant for those of you preparing for the NEET examination. As you know, genetic disorders encompass a wide array of conditions that result from anomalies in an individual's DNA, whether due to mutations in a single gene, abnormalities in chromosomes, or more complex multifactorial causes. Today, I will be walking you through the classification of genetic disorders, their molecular basis, examples of key monogenic and chromosomal disorders like thalassemia, sickle cell anemia, and Down syndrome, and how they are inherited. We'll also touch upon mitochondrial inheritance and some of the latest clinical and diagnostic tools used in detecting and managing these conditions. NEET aspirants, I will be marking out high-yield facts that are frequently tested, so make sure to take notes when I highlight those NEET points. You will find that understanding genetic disorders not only strengthens your knowledge in inheritance and molecular biology, but also bridges the gap between basic science and clinical medicine, something that is crucial as you advance in your studies or prepare for medical entrance tests. Let's begin. Before diving into the content, let's take a quick look at the objectives of today's lecture. Our goal is to achieve a thorough understanding of the molecular and chromosomal basis of genetic disorders. This includes identifying whether a disorder results from a mutation in a single gene or a large-scale chromosomal abnormality, and how those molecular changes translate into clinical symptoms. We will start by classifying the different types of genetic disorders: monogenic or Mendelian disorders, chromosomal disorders, multifactorial disorders, and mitochondrial disorders. This classification is essential, not only from a theoretical standpoint but also in clinical diagnosis. Next, we'll analyze case studies of key genetic conditions like thalassemia, sickle cell anemia, Down syndrome, Turner syndrome, and hemophilia. For each, we'll examine the genetic mutation involved, inheritance pattern, phenotypic presentation, and diagnostic strategies. In addition, for each disorder, I will call out high-yield NEET pointers so that you're equipped with the most testable facts for your entrance exam. NEET often tests chromosomal number, gene loci, inheritance type, and hallmark symptoms, so we'll cover those in detail. We'll also discuss pedigree analysis, an important tool for tracking inheritance patterns across generations. By the end of this session, you should be able to interpret pedigrees and classify them as autosomal dominant, autosomal recessive, or X-linked. Finally, we will briefly touch on modern genetic tools like gene therapy and CRISPR-Cas9 editing that are becoming increasingly relevant. By the end of this lecture, my aim is for you to not only memorize but deeply understand how mutations at the molecular level manifest as clinical syndromes and how they are passed down generations. This foundational slide breaks disorders into four main types: monogenic, chromosomal, multifactorial, and mitochondrial. Understanding these categories is crucial for determining the origin and mechanism of each condition. Monogenic disorders, also known as Mendelian disorders, are caused by mutations in a single gene. These disorders follow Mendelian inheritance patterns, meaning they can be autosomal dominant, autosomal recessive, or X-linked. Examples include thalassemia, sickle cell anemia, cystic fibrosis, and hemophilia. These are frequently tested in NEET, so make sure you memorize the gene involved and its chromosomal location. Next, we have chromosomal disorders, which result from changes in chromosome number or structure. These are typically much more severe and may affect multiple systems. Classic examples are Down syndrome, trisomy 21, Turner syndrome, monosomy X, and Klinefelter syndrome, XXY. These disorders arise primarily from errors during meiosis, such as non-disjunction. Multifactorial disorders are caused by the combined effects of multiple genes and environmental factors. Examples include common conditions like type 2 diabetes, hypertension, and certain cancers. These are more complex and don't follow simple Mendelian patterns. Finally, mitochondrial disorders are passed down exclusively through the maternal line because only the egg contributes mitochondria to the embryo. These disorders affect high-energy systems like the brain, muscles, and eyes. An example is Leber's hereditary optic neuropathy, LHON. Pushpin NEET tip here. Most NEET questions focus on monogenic and chromosomal disorders. You should be able to answer questions like, What gene is mutated in sickle cell anemia? Or, what is the chromosomal basis of Down syndrome? Focus on gene location, inheritance pattern, and phenotypic presentation. Now, let's dive deeper into monogenic disorders, which are perhaps the most straightforward to understand and frequently tested in NEET and university exams alike. Monogenic disorders arise due to mutations in a single gene. These mutations can occur in coding regions, introns, or even regulatory sequences that affect gene expression. These disorders follow Mendelian inheritance patterns, which you should already be familiar with from previous chapters. Specifically, the inheritance can be autosomal dominant, where only one mutant allele is sufficient for the disorder to manifest. Example, Huntington's disease. Autosomal recessive, where both alleles must be mutated. Classic examples include sickle cell anemia and thalassemia. X-linked, where the gene involved is located on the X chromosome. These can be dominant or recessive, though X-linked recessive disorders like hemophilia A are more common. In monogenic disorders, the penetrance is usually high, meaning that if an individual has the disease-causing alleles, they are very likely to show symptoms. The age of onset is often early, frequently in infancy or childhood, because the mutation usually affects fundamental processes like hemoglobin synthesis or enzyme function. From a molecular perspective, these mutations can be point mutations, single nucleotide substitutions, insertions, deletions, or even splice site mutations. For example, in sickle cell anemia, a point mutation causes a change in one amino acid in the hemoglobin beta chain, leading to a drastic phenotypic effect. Finally, note that although these disorders involve just a single gene, the effects can be systemic and complex due to the phenomenon of pleiotropy, where one gene influences multiple traits, and codominance, where both alleles are expressed, as seen in carriers of sickle cell who have traits from both normal and mutated alleles. Let's begin our case studies with thalassemia, a classic example of a monogenic, autosomal recessive disorder that affects hemoglobin synthesis. This is both highly testable in NEET and critical for clinical practice. Thalassemia is caused by mutations in the genes responsible for synthesizing the alpha symbol, or beta symbol, globin chains of hemoglobin. Hemoglobin, as you know, is the oxygen-carrying protein in red blood cells made of 2 alpha and 2 beta chains in adults. Alpha thalassemia is caused by deletions or mutations in the HBA1 or HBA2 genes located on chromosome 16. Beta thalassemia, more common in India and Mediterranean populations, results from mutations in the HBB gene on chromosome 11. The result? Inadequate globin chain production leads to abnormal hemoglobin, which in turn causes ineffective erythropoiesis, anemia, and destruction of red blood cells in the bone marrow and spleen. Clinically, patients present with severe anemia, bone deformities due to marrow expansion, hepatosplenomegaly, enlarged liver and spleen, growth retardation in children. Diagnosis is done using electrophoresis of hemoglobin, genetic testing, or blood smear. Carriers are usually asymptomatic but can pass the defective allele to offspring, making carrier screening important in regions with high prevalence. Also, note the spectrum of disease severity. Thalassemia minor, one allele affected, usually asymptomatic. Thalassemia major, both alleles affected, severe disease. Thalassemia intermedia, moderate form. From a genetic standpoint, this is a good example to understand compound heterozygosity, where 2 different mutations in the same gene can cause disease. Now we turn to sickle cell anemia, another autosomal recessive monogenic disorder. This one's particularly important because it illustrates a point mutation causing a profound structural change in the hemoglobin protein. The mutation is a single base substitution in the HBB gene on chromosome 11, where the codon GAG, which codes for glutamic acid, is mutated to GG, resulting in valine replacing glutamic acid at the 6th position of the beta-globin chain. This small change causes hemoglobin to polymerize under low oxygen conditions, distorting red blood cells into a characteristic sickle shape. These sickled cells are fragile, leading to hemolysis, blocks small blood vessels, causing painful crises. shorten lifespan of RBCs, leading to anemia. Clinically, patients present with chronic fatigue and pallor, painful episodes, vaso-occlusive crises, increased risk of infections and stroke, splenic and skeletal complications. Interestingly, individuals who are heterozygous for the mutation, carriers, have a selective advantage in malaria-endemic regions because the sickled cells do not allow the malaria parasite to complete its life cycle. NEET tip: remember the mutation, DAGGTG on chromosome 11, and the condition is autosomal recessive. It's also a classical example of pleiotropy, where one mutation affects multiple systems and codominance because carriers show traits of both normal and mutant hemoglobin. Diagnosis is via hemoglobin electrophoresis, HPLC, or genetic testing. Treatment is mostly supportive hydroxyurea, pain management, and in some cases, bone marrow transplant. This case elegantly ties molecular biology with clinical presentation, showing how a single base change can impact the entire body. Now we move into chromosomal disorders, which are due to large-scale abnormalities in chromosome number or structure. Unlike monogenic disorders, these do not involve mutations in single genes, but rather the addition, deletion, or rearrangement of whole chromosomes or large sections of them. There are two main types. Numerical abnormalities. These are known as aneuploidies, such as trisomy, extra chromosome, or monosomy, missing chromosome. Structural abnormalities. These include deletions, duplications, translocations, and inversions. These disorders are typically caused by errors during cell division, particularly during meiosis, when chromosomes are supposed to separate evenly into gametes. A failure in this process, called non-disjunction, leads to an abnormal number of chromosomes in the zygote. Because entire chromosomes are involved, the effects are usually multisystemic and severe. Most chromosomal abnormalities are incompatible with life, and those that are viable typically lead to developmental delays, facial dysmorphisms, and organ malformations. Some of the most well-known chromosomal disorders include Down syndrome, trisomy 21, Turner syndrome, monosomy X, Klinefelter syndrome, XXY, Patau and Edwards syndromes, trisomies 13 and 18. Pushpin NEET tip: non-disjunction is the primary cause of aneuploidy. Expect questions like, which stage of meiosis is affected in Down syndrome? Or, what is the karyotype in Turner syndrome? Be sure to memorize the chromosome numbers and clinical features associated with each disorder. These are typically diagnosed by karyotyping, which provides a full chromosomal profile and may be supplemented by FISH or array CGH for smaller changes. Alright class, let's dive into a classic example of a chromosomal disorder, Down syndrome. You've probably heard of it before, but let's understand what's really happening at the genetic level. So, Down syndrome is caused by an extra copy of chromosome 21. Normally, we all have 46 chromosomes, 23 from each parent. But in Down syndrome, there are 47 because chromosome 21 appears 3 times. We call that trisomy 21. Now, why does this happen? Most often, it's due to something called non-disjunction during meiosis. Basically, the chromosomes don't separate properly. This usually occurs in the egg, especially in older mothers. There are a few types. Free trisomy, which is most common, Robertsonian translocation, where part of chromosome 21 gets attached to another chromosome, and mosaicism, where only some cells have the extra chromosome. What do we see clinically? You might recognize some key features: a flat facial profile, upward slanting eyes, a single crease across the palm called a simian crease, and developmental delays. They also often have heart defects, particularly atrioventricular septal defects. An ED tip here. Remember, chromosome 21, 47 chromosomes, and the word non-disjunction. Those are favorites on MCQs. Also, they might ask you which parent usually contributes the extra chromosome. Answer: usually the mother, due to age-related risk. Diagnosis? Prenatal screening like ultrasound, triple test, and amniocentesis. Then we confirm with karyotyping. Alright class, now that we've covered Down syndrome in depth, let's explore some of the other important chromosomal disorders you need to know, especially for NEET and your university exams. Let's start with Turner syndrome, which we already mentioned a bit earlier. This is the only viable monosomy in humans, remember that. Affected individuals have only one X chromosome, so 45 chromosomes total.
Noi chromosome.
These individuals are genetically female, but they present with short stature, delayed puberty, and infertility because of streak ovaries. You might see features like a webbed neck, a broad chest, and even congenital heart defects like coarctation of the aorta. NEET tip. Turner is X0, female phenotype, and often shows primary amenorrhea. Next up, Klinefelter's. This one is extra X in males, so 47 chromosomes, with an XXY karyotype. These individuals are genetically male, but the extra X leads to feminization features. They tend to be taller than average, have reduced facial and body hair, gynecomastia, that's breast development in males, and infertility due to underdeveloped testes and low testosterone. Think of this as the opposite end of the sex chromosome imbalance spectrum compared to Turner. NEET angle. XXY equals male, tall stature, gynecomastia, infertility. Remember, bar bodies are present because of the extra X. Now we move into autosomal trisomies again. Patau syndrome is trisomy 13. This is a severe chromosomal disorder with low survival beyond infancy. The hallmarks? Microcephaly, small head, holoprosencephaly, brain doesn't divide properly, cleft lip and/or palate, polydactyly, and congenital heart defects. Babies born with this often have multiple organ system issues, and sadly, survival is usually less than 1 year. Finally, we've got Edwards syndrome, or trisomy 18. Like Patau, this is also a severe autosomal trisomy, and again, life expectancy is very short. The classic findings are clenched fists with overlapping fingers, rocker bottom feet, micrognathia, small jaw, and severe intellectual disability. You'll also see congenital heart defects and sometimes growth restriction in utero. A big diagnostic clue? The hand position. The clenched fists are very characteristic and often seen on prenatal ultrasound. NEET clue. Trisomy 18 equals election age. Think of someone clenching their fists in political frustration. It's a silly memory aid, but it sticks. Also note, trisomies get worse as the number gets lower in terms of severity. Trisomy 21 Down syndrome is most survivable, trisomy 18 Edwards is worse, and trisomy 13 Patau is the most severe. That's not always perfect, but it's a useful trend to remember. What is pedigree analysis? It's essentially a family tree with genetic clues. It's how we trace how a particular trait or disorder is inherited through generations. Let's decode the basics first. Small blue diamond, understanding pedigree symbols. So here's the good news. Pedigree charts use standardized symbols, so once you learn them, they don't change. Males are squares, females are circles. If the shape is shaded, that person is affected. Unshaded equals unaffected. A horizontal line between a male and female equals marriage or mating. A vertical line coming down equals offspring. If siblings are connected by a horizontal line, they're from the same parents. Now, if you see a half-shaded shape, it usually means a carrier, especially in X-linked recessive or autosomal recessive disorders. Sometimes you'll also see a double horizontal line between partners. That represents consanguinity, meaning the parents are related, like cousins. Small blue diamond decoding the inheritance pattern. Now comes the detective work, looking at the pedigree and figuring out what type of inheritance is at play. Let's go through the major types you'll need to recognize. 1. Autosomal dominant. Appears in every generation. No skipping. Affected individuals have at least one affected parent. Males and females are equally likely to be affected. It's passed to both sons and daughters. Brain example, Huntington's disease. Backhand index pointing right, NEET clue. If it's showing up in every generation and affects both sexes equally, go for autosomal dominant. 2. Autosomal recessive. Usually skips generations. Often seen when both parents are carriers. More likely in consanguineous marriages. Affected person can be born to 2 unaffected carrier parents. Brain example, cystic fibrosis. thalassemia. Backhand index pointing right, NEET clue. If healthy parents have an affected child, think autosomal recessive. 3. X-linked recessive. More males are affected because they only have one X. Females are usually carriers. An affected male can't pass it to his sons, but all daughters become carriers. Can skip generations. Brain example, hemophilia, Duchenne muscular dystrophy. Backhand index pointing right, NEET clue. If the disease jumps a generation and mainly affects males, it's probably X-linked recessive. 4. X-linked dominant. Affected mothers can pass it to sons and daughters. Affected fathers pass it only to daughters, never to sons. Usually no skipping generations. Brain example, Rett syndrome. Backhand index pointing right, NEET clue. If every daughter of an affected father is also affected, it's X-linked dominant. 5. Mitochondrial inheritance. Passed only from mother to all children. Fathers never pass it on. Affected mothers equals 100% of kids affected. Brain example, Leber's hereditary optic neuropathy. Backhand index pointing right, NEET clue. If only the mother is transmitting it and all offspring are affected, it's mitochondrial. Let's talk strategy, especially for NEET. Check sex distribution. Are mostly males affected? Might be X-linked. Look for generation skipping. If yes, think recessive. Every generation affected? Likely dominant. Only mother passing it on? Consider mitochondrial. It's like playing a game. Every chart is a puzzle. And once you practice 10 to 15 of them, you'll start seeing patterns automatically. Alright everyone, time to shift gears. We've done the deep dives, we've explored the clinical tools, and we've built up a strong conceptual foundation. Now let's kick into NEET revision mode. This slide is your golden ticket, your high-yield snapshot of everything genetic disorders related that NEET loves to test. I want you to think of this as your cheat sheet, your MCQ ninja board, your last-minute power grid when you're revising the night before your exam. Okay, class, now we're entering a really exciting part of genetics, clinical techniques. This is where we step out of the textbook and into the real world of genetic diagnosis and research. You've learned about the disorders. You've understood inheritance. But how do we actually detect, analyze, or confirm these genetic conditions in the lab or clinic? Let me take you on a tour of the most important tools we use. I'll break them down into what they are, how they work, and what you need to remember for NEET and university exams. DNA 1. Karyotyping. Let's start with the classic, karyotyping. Imagine you've got a big jigsaw puzzle of all 46 human chromosomes. Karyotyping is how we lay those pieces out, line them up in order, and look for missing, extra, or broken parts. We usually arrest cells in metaphase, stain them, and photograph the chromosomes. Then we arrange them into a neat display called a karyogram. Eyes, what can we detect? Aneuploidies like Down syndrome, trisomy 21. Turner syndrome, 45X. Klinefelter's, 47XXY. Backhand index pointing right, NEET tip. If the question mentions a full view of chromosomes, it's talking about karyotyping. Petri dish 2. Amniocentesis and chorionic villus sampling, CVS. These are prenatal techniques. Think of them as sampling the baby's environment to check for genetic issues before birth. Amniocentesis. We take a bit of the amniotic fluid around the fetus, usually after 15 weeks. CVS. We take a sample from the placenta, chorionic villi, done earlier, around 10 to 12 weeks. Both allow us to collect fetal cells, which we then test via karyotyping or even molecular methods like PCR. Backhand index pointing right, NEET tip. CVS is earlier but riskier. Amniocentesis is later but safer. Test tube 3. Fish fluorescence in situ hybridization. Now, this one's cool. Fish uses fluorescent DNA probes to bind to specific chromosome regions. When viewed under a special microscope, the glowing spots tell us if a specific gene or chromosome piece is present or missing. It's faster than karyotyping and can detect microdeletions or translocations that are too small to see on a regular karyotype. Brain example, diagnosing DeGeorge syndrome, 22q11 deletion. Backhand index pointing right, NEET clue. If the question says fluorescent tags used to detect genetic abnormalities, think fish. DNA 4. PCR, polymerase chain reaction. You've probably heard this one during COVID testing, right? PCR is like a DNA photocopier. It amplifies even tiny amounts of DNA. It's especially useful for detecting specific gene mutations, like those in thalassemia, sickle cell, or CFTR gene in cystic fibrosis. We can even use PCR for carrier screening, forensic testing, and infectious disease detection. Backhand index pointing right, NEET tip. PCR equals detect point mutations, amplify DNA. Test tube 5. Gel electrophoresis. Imagine you've got a bunch of DNA fragments of different sizes. Gel electrophoresis is how we separate them by running them through a gel using electricity. Smaller fragments move faster. Bigger ones lag behind. The pattern you get is called a DNA fingerprint. Uses? Forensics. Paternity tests. Checking the results of PCR. Backhand index pointing right, NEET clue. If they mention separating DNA fragments by size, it's gel electrophoresis. DNA 6. DNA sequencing. This is the holy grail of genetic testing. We actually read the entire genetic code. It's how scientists discover disease-causing mutations in BRCA1, BRCA2, and many others. Although it used to be very expensive and slow, today's next-gen sequencing, NGS, is much faster and cheaper. This is what we use in whole genome or whole exome sequencing for patients with undiagnosed genetic disorders. Backhand index pointing right, NEET tip. May not be directly asked often, but useful for understanding advanced applications. Let's talk about some of the cutting-edge advances in genetics that are really changing the game when it comes to genetic disorders. These aren't just textbook stuff anymore. These are real technologies and treatments making a difference today and in the near future. 1. Gene therapy. First up, gene therapy. This is where we actually insert healthy copies of a gene into a patient's cells to replace or fix faulty ones. It's like giving cells a fresh instruction manual. Currently, there are promising clinical trials for conditions like beta thalassemia, a serious blood disorder, and SCID, severe combined immunodeficiency, which is often called the bubble boy disease. These trials aim to correct the underlying genetic defect, not just manage symptoms.
2. CRISPR-Cas9.
Next, you've probably heard about CRISPR/Cas9. It's a powerful genome editing tool. Imagine having molecular scissors that can cut DNA at a precise spot. Scientists use CRISPR to edit faulty genes, potentially curing inherited diseases. It's fast, efficient, and cheaper than older gene editing methods. CRISPR is still in early clinical stages for many disorders, but it's showing incredible promise, especially for blood disorders like sickle cell anemia. 3. Carrier screening. Now, this one is especially important in countries or communities where consanguineous marriages between close relatives, are common. Carrier screening involves testing individuals to see if they carry genes for certain inherited disorders, even if they're healthy themselves. This helps identify risks before having children, allowing for informed decisions and sometimes preventive measures. 4. Mitochondrial replacement therapy. Finally, mitochondrial replacement therapy is a fascinating advance for disorders passed down from the mother's mitochondria, the tiny powerhouses in our cells. In some diseases, faulty mitochondria cause serious problems. This therapy allows replacing defective mitochondria with healthy ones from a donor. All right, team, we've covered a lot today, so let's take a moment to wrap it all up with a quick summary, kind of like hitting the pause button and looking at the big picture. What did we learn? Genetic disorders arise because of changes or mutations in our DNA. These can be single gene mutations, chromosomal abnormalities, or multifactorial issues. We went through types of genetic disorders: autosomal dominant and recessive, X-linked, mitochondrial, and chromosomal disorders like Down, Turner, and Klinefelter syndromes. Each has its own pattern and unique features. Knowing these helps with diagnosis. We explored how pedigree analysis is a powerful tool to track inheritance patterns in families. Think of it as your detective's map for genetic clues. Then we discussed various clinical techniques like karyotyping, PCR, FISH, and newer DNA sequencing methods that help us detect and understand these disorders at the molecular level. We drilled down on the NEET high-yield points, chromosome numbers, inheritance modes, hallmark symptoms, because that's what you'll see in exams, and it's the best way to secure those marks. Finally, we peeked into the future with recent advances like gene therapy, CRISPR, carrier screening, and mitochondrial replacement therapy, all showing how genetics is evolving from just diagnosis to real treatment and prevention. Why does this matter? Because genetics is no longer just academic, it's changing lives. From helping families understand risks, to designing personalized treatments, to potentially curing disorders we thought were untreatable, this field is at the heart of modern medicine. Now we've reached the end of our lecture on genetic disorders. Before we wrap up completely, here are the key references and sources I used to prepare this material. These include trusted textbooks, recent research articles, and reliable academic websites. Great resources if you want to dive deeper. I highly encourage you to explore these references to strengthen your understanding and stay updated. Most importantly, thank you so much for your attention and participation throughout the lecture. It's been a pleasure guiding you through this complex but fascinating topic. If you found this lecture helpful, don't forget to subscribe to the channel. That way, you won't miss out on any upcoming lectures on genetics or other biology topics tailored for NEET and university students. Feel free to drop your questions or topic requests in the comments. I love hearing from you, and it helps me make better content. Stay curious, keep studying, and I'll see you in the next lecture.